NAP,
NAP.
SIS.
Okay. So what is the one word summary that we did for diplotin? Tell me what happens in diplotin. Can you see the screen?
Yes, sir.
Just let me make it big. Yeah. So what happens in diplotin? The one line summary.
Chiasmata is visible.
Chiasmata. And what was chiasmata? Remember, there were two. If there were two chromosomes like this,
Yes.
then this thing, this crossing over, was called chiasmata, right?
Yes.
So this same, like these two chromosomes, basically. Now read the sentence with synapses. What it says. The sentence that.
Of synapses.
So they are together joined. These two chromosomes are joined together. Okay. Then they start dissolving, and this joining is called synapses. Then they start dissolving, but they stay connected just at this one point. Okay. That's called chiasmata. You understand?
Yes.
And then they break from that point also, an exchange of genetic material. So part of one chromosome goes to the other, and the part of other chromosome comes to the first one. Understood?
Yes.
Okay. Very well. So shall we go with some questions?
Yes.
Hiram, are you ready?
Yeah.
Okay, very well. So let me share the screen. Yes, can you see the screen, both of you? And Aisha, is your enter key working today? Yes, no?
Yes, sir.
Yeah, it is. Oh, very good. Great. So yeah, so you can enter your answers. Good, good, very good. So glad to hear that. Okay, so let's start with some very basic ones. Let's start with question 1. Let's see, asked in 2017 NEET. So let's start with Aisha. Aisha, you ended yesterday, so let's start with you. The good luck continues. So the same thing, you can read and then you can answer. In the meanwhile, Iram can register her answer in the chat. Okay, you can answer as fast as you want. Aisha, are you there?
Yes, sir.
Yeah, let's do it.
Which of the following options gives the correct sequence of events during mitosis? Condensation, nuclear membrane, disassembly, arrangement at equator, centromere division, segregation, and telophase.
Yeah, so this is the, no, you don't have to read all. So you just know that they will just tweak with the sequence. So correct sequence of events. Condensation means chromosomal condensation. So chromosome condenses, then the nuclear membrane disassembly means it disappears. Arrangement at the equator means all the chromosomes come at the equator. Centromere division is when it's basically, so they are not using words like prophase, metaphase, anaphase, but just giving you characteristics. Centromere division is when during anaphase they are pulled apart. Segregation is when they move towards each side, and telophase is the last. So tell me, which do you think is the right sequence? A, B, C, or D? Aisha, you have to speak.
Sir, option A.
Yes, very good. See, we started with the right answer, and Iram gave a lightning fast right answer. I think Iram didn't even read other options, in my opinion. Did you read? Like, it's not a good thing. Did you read other options?
No.
See, I just got, because the moment I asked what is the right answer, you're like, hey, you're so confident. Yeah, probably you looked at condensation and telophase. Okay, but you should also read other options. Okay, you have 1 minute. Don't worry. It's better to take 30 seconds instead of giving wrong. But this time it was right. Well done. Okay, luck favors the brave. Okay, Iram, your turn. Question 2.
Anaphase promoting complex is a protein degradation machinery necessary for proper mitosis of animal cell. If APC is defective in human cell, which of the following is expected to occur? A. Chromosomes will be fragmented. B. Chromosomes will not segregate. C. Recombination of chromosomes arms will occur. D. Chromosomes will not condense.
Yeah. Now, do not get intimidated by the question. Things like these, which are not in NCERT, they will give you explanation. Anaphase promoting complex. Now, let me explain this question why I've taken this, because there's no mention of anaphase promoting complex, right? It is a protein complex, and such complexes are present during each phase. There are many proteins that help a cell to divide basically and go into the cell cycle, So there are lots of proteins, all the names you cannot remember, but see, they're telling you. Now, what happens is an average student will just read this question and will just give up and realize, okay, I have not studied about this. I don't know what it is. But a Rahul Kushwaha student will do this thing. He or she will read the complete question without getting intimidated by the question. Try to come to the bottom line. The question is simple. Anaphase promoting complex, whatever it is, we don't care, is a protein degradation machinery necessary for proper mitosis of animal cells. Again, protein degradation machinery. So if it degrades some proteins. Now, we know that the cell division, the prophase, metaphase, anaphase, telophase is all about karyokinesis. This you know? Yes or no?
Yes.
Yes or no? It's all about karyokinesis, about the nucleic acid, about DNA. So if it is a protein degradation machinery, what proteins it might be degrading, which are helping anaphase to proceed, right? You understand?
Yes.
So if APC is defective, then in a human cell, if anaphase promoting complex, look at the name, anaphase promoting complex, it promotes anaphase. So if it is defective, without even knowing what it does, it may not be able to promote anaphase now. So the question is asking a simple thing. If you do not allow anaphase to happen in a human cell, then what will happen? Which of the following is expected to occur? Instead of asking a simple question that if anaphase does not happen, what will be the consequence? They are giving you a whole story to just frighten you and leave this question, so because negative marking is there, but see if you can understand. Okay, do you understand the question now? This is also a part of the training in the class that is done. So if anaphase promoting complex is defective, then anaphase is not happening. So what will happen? What do anaphase do basically in one line? That's not the right answer, by the way, because focus on what anaphase is. What is anaphase? Where does it come? Prophase, metaphase, then comes anaphase. What happens in anaphase?
Splitting of.
What is this? Segregation of chromosomes, right? Then why have we chosen chromosome will not condense? Condensation has nothing to do with anaphase, right? What will be the right answer?
B.
B. You understand, Aisha, why B is the right answer? Howsoever difficult language they are giving you. Now you will see anaphase promoting complex. What is this? And that is what they exactly wanted to do. So to make a entrance difficult, they want to confuse you. If you get confused, then you don't deserve to do good in the entrance and you will not be getting a good rank. Everyone knows about cell. So how to make entrances competitive? By just doing this, exactly this. So you just have to focus on the bottom line. The bottom line is if APC is defective, promotion of anaphase will not happen. So anaphase, without anaphase, chromosome will not segregate. Okay. Chromosome will not be fragmented. There is no fragmentation of chromosome, right? In any cell division phase. We never break chromosomes. Yes or no? So A is an absurd option. Recombination of chromosome arms will occur. This happens in packaging of prophase. Remember? Recombination happens in prophase. Remember, Aisha and Iram?
Yes.
So that also has nothing to do with anaphase. Chromosome will not condense. Chromosomes do not condense once they have once they are out. So chromosomes have already condensed in prophase. Remember? Before you leave your house, you get ready and condense and pack things, right? If you are going somewhere, you pack things, which means you condense your things. You fold all your clothes, you keep your books arranged properly in a suitcase. That suitcase size is very less and everything is compact and packed. Then you leave your house. So when chromosomes leave nucleus in prophase, at the beginning of metaphase only, before that they start condensing. So then when the nuclear membrane will dissolve at the end of prophase and in the beginning of metaphase, they will come out of it in a compact manner. So no compaction or no condensation happens afterwards. Okay. Anaphase is very late. Till then, they are all condensed already. So which is the only option that makes sense? B. Was it a difficult question or an easy question? Iram.
Yeah, easy.
It was an easy question packed in a different, like in a difficult language, right? So you just have to figure out the question. Don't get scared by the language. Okay, Aisha. DNA applications. Question 8, you all know, I just told you about. What is the answer for question 8? Quickly. This is a very, very frequently asked question. Aisha, will you read the question? Aisha, are you there?
Yes, sir.
Yeah.
In meiosis, crossing over is initiated at option C.
Of course, packaging of prophase. If these options were not there, if prophase, metaphase, anaphase, telophase were there, you will just write prophase. Very good. So fast, even faster than lightning speed. Question number 10. Hiram. No, okay, Aisha. That was easy one. Okay, let me give Aisha also equal opportunity. Some difficult questions. Aisha, question 10. Hiram, you can give the answer in the chat quickly if you want.
A somatic cell that has just completed the S phase of its cell cycle as compared to gamete of the same species has twice the number of chromosomes and 4 times the amount of DNA, 4 times the number of chromosomes and twice the amount of DNA, twice the number of chromosomes and twice the amount of DNA, same number of chromosomes but twice the amount of DNA.
What do you think, Aisha? When I do this with students, I feel like I'm playing who will become a millionaire, kaun banega crorepati type things. And I wish, yeah, your answer is correct and lightning fast. Are you reading other options as well? This is my worry. You know. Now
Yes,
I'm getting.
I did read.
Now I'm getting worried about Iram's obsession of being so fast, and she can actually miss other options.
Sir, option
Aisha, yes, very good. Aisha, again, you did it in like good time, because if you just do it like this, you will have a good entrance. You did it within 1 minute, but it was correct. I'm happy that both of you gave correct answer. Okay, it's option A. Why? Can you explain? Why do you think it's option A? Option is correct, but you have to just justify it. If you know the answer. Iram, justify.
It's because the DNA content of the cell doubles during the S phase.
So
While the number of chromosomes remains same.
yeah, so the somatic cell that has just completed the S phase of its cell cycle as compared to gamete of the same species has twice the number of chromosomes and 4 times the amount of DNA. So gamete is haploid. This is the justification. Gamete is haploid and a somatic cell is diploid. So it is already 2n and this is n, the number of chromosomes. Okay. After the completion of S phase, a somatic cell still will be 2n. So then twice the number of chromosomes, n and 2n. But the DNA content is now double. So DNA content is 4n, like 4 times. You understand? Aisha? Do you understand? Can I get a whiteboard here? Okay, very good. I got a thumbs up. Good. Okay, so for Iram, there's one more question like this, which I have figured out. So it also has this book, like the book I'm referring to also has questions, which was before like NEET. Earlier it used to be AI PMT, and in that the syllabus was also out of NCERT. A lot of things were asked from out of NCERT. Now that thing is not, they don't do that for good. They stick to NCERT mostly, but there are some concepts that they ask from outside of the NCERT, and that I will tell you right away whenever we will do that, those concepts. Okay, question number 30. Quick. This is
in somatic cell cycle. Yeah, in somatic cell cycle, A in G1 phase DNA content is double the amount of DNA present in original cell. B, DNA replication takes place in S phase. C, a short interphase is followed by a long mitotic phase. D, G2 phase follows mitotic phase.
And what do you think is the right answer? Aisha, you can write in the chat. Prove that your enter key is working. Irami. Okay. Oh, okay. Aisha, what's your answer? Great. Both of you, right answer. I realize one thing, both of you are very peculiar students. Both of you are brilliant with concepts and very bright students. Iram is like that power play player, like to just boom, boom, shots and shots. Aisha is like a test match player. She reads, she realizes, okay, I don't have a margin of error. But ultimately both of you give right answers. So that's good. Like me personally, I'm like more Aisha kind of, I was Aisha kind of student. It takes guts to be Iram kind of student. Yeah, but be careful, okay, Iram? Okay. Aisha, question number 32. Most of the questions in cell division chapter is concept based. Aisha, question 32.
Which one of the following
Precedes, not proceeds. Precedes.
Oh,
precedes mean which comes before. Yeah.
Reformation of the nuclear envelope during M phase of the cell cycle, decondensation from chromosomes, and reassembly of the nuclear lamina, transcription from chromosomes and reassembly of the nuclear lamina, formation of the contractile ring and formation of the phragmoplast, formation of the contractile ring and transcription from chromosomes.
Yes. So why I ask you to like just try it, but there are a few things I want to tell you. This question is from NCERT only, okay? But none of these words like phragmoplast or contractile ring you have studied, right? But still this question can be asked in simple words. So what do you think? Let's just make a guess before I dictate you things about it. you can still figure out the right answer. Look, you can understand A, B, and C at least, right? A, B, and D. Okay, let's keep this question on hold. Let me dictate you what is phragmoplast. Write down phragmoplast. Write it somewhere with your notes. This word is not there in NCERT, but this concept is. Phragmoplast is a structure in plant cell. Phragmoplast is a structure in plant cell that forms during late cytokinesis. That forms during late cytokinesis and helps. Remember, in plant, it is inside out cell wall formation because you cannot pinch in cell wall.
Yes.
Yeah. So to make things inside out, there has to be something. So if I start making a wall in the center of the room and I'm extending it to merge it with the already present walls. So by the time it has not joined the already present walls, it is just floating. So imagine a cell. A cell is like a fluid filled chamber, right? So this wall is present in a fluid filled chamber. So something has to keep that wall in position before it joins to the actual side walls to make a new cell wall, right? So these phragmoplasts are nothing but phragmoplast microtubule filaments which hold this wall, which is forming from the center and going towards the side walls in place. So that's phragmoplast. And this is very much important. This is not present in animal cell because in animal cell, you pinch the cell membrane from out to in. Okay, in plant, you make it inside out. So you need to have something which can keep this in place till it is completing. So that's phragmoplast. So write down, it helps in cell plate assembly. In cell plate assembly during new cell wall formation. It helps in the cell plate assembly. It helps in holding the cell plate assembly during new cell wall formation. Nuclear lamina, like these, all these words are some difficult technical words, but the concepts are in NCERT only. Decondensation of chromosomes is, yes, Iran.
My answer is correct?
Oh, I have not seen the answers yet. No, no one's answer is correct. That's why I'm first explaining you these terms, okay? So decondensation of chromosome is clear to you? Reassembly of nuclear lamina. Lamina means the membrane. Write down. Nuclear lamina means nuclear membrane. First, understand this question. That's what I'm saying. Nuclear lamina is nuclear membrane. Formation of contractile ring. Now, what is contractile ring? Write down. Contractile ring. So it helps. It is the. So when cell is dividing during cytokinesis, so when it pinches from outside in, how does it pinch? How can you pinch something outside in? You make a ring around it and keep contracting it. So this is, so let's say this is the, my finger width is the width of or the diameter of a cell, okay, on the outside. Is it clear? Can you see it? And if I just keep contracting it over time, I'm actually pinching the cell. take a balloon, fill it with water, fit it in your fingers and keep contracting it. So what are you doing? You are doing cytokinesis. You understand?
Yes.
That's contractile rate. So basically it's nothing but they're talking about cytokinesis. So all the words are clear to you. Now tell me which is the answer. Read and tell me. If you give right answer to this, both of you, then it means you have understood the whole cell division cycle very, very clearly. Read the question again if required and give me the answer. Take some time, no problem. Focus on the word precedes. Precedes means which happens before. Succeeds means which happen after.
Sir, what is the meaning of transcription?
Okay. So there are three things. Replication is making DNA from DNA. We studied central dogma, remember? Central dogma of biology. Did I discuss this with you in chapter 8, cell?
No.
Yes or no? No? Okay.
No,
So
sir.
replication is DNA making DNA from DNA. Transcription is making RNA from DNA. And translation is making protein from RNA. Just write it down. Replication, synthesis of new DNA from old DNA. So replication happens in S phase, right? Then transcription is making of RNA from DNA. And translation is making proteins from RNA. So this is one of the toughest question that can be asked. If you understand this, cell cycle is sorted. So first replication happens, then transcription, then translation. But you have to think when is RNA synthesized, when is protein synthesized, and when is DNA synthesized in a cell cycle. Okay, let me take you through the right answer. Question is, which one of the following, A, B, C, D, precedes, which means happens before reformation of nuclear envelope during M phase of the cell cycle? So now we don't have to worry about M phase, because in M phase we know that reformation of nuclear envelope happens in the telophase, right? Yes or no? Yes or no? During the telophase, when they reach towards the end, the nuclear membrane starts reforming as two daughter nuclei. DNA has come to the poles. Now there will two nucleus, and then cell division will happen in the center. Are you following me, Iram and Asha? Are they on the
Yes,
same
sir.
page? Yeah. So the question is simply asking what's happening before M phase. So what is before M phase? before M phase, which phase is there in the cell cycle? This is what you have. This is what the question is asking.
G2.
Exactly. And what used to happen in G2? What happens in G2?
Synthesis.
Synthesis of proteins, right? Yes or no?
Yes.
Protein synthesis were there in G2. But the question has one catch here. Pay attention. Can you see my cursor? If I can move. Can you see this cursor if I'm moving this?
Yes. Yes.
So this reformation of the nuclear envelope during M phase happens in telophase. Yes. What happens
Yes.
just before telophase? Which phase comes just before telophase?
Anaphase.
And what happens in anaphase? What happens in anaphase?
Chromosomes are pulled to opposite poles.
They start coming to the opposite pole, and at the beginning of telophase, you start seeing a contraction. Remember, I made a cell with a furrow at both the ends at the beginning of telophase, right? So the
Yeah.
contractile ring starts functioning there. And so contractile ring is for animal cell, and phragmoplast is for plant cell. Okay. So the right answer to question number 32 is option C. Just before the nuclear envelope reappears, like is reformed, the cell starts contracting to do cytokinesis, right? Yes or no? Other options don't make much sense because they are happening after it or very much before it. Decondensation of chromosomes happen after, right? Or almost at the same time when the nuclear envelope is reforming during telophase. Decondensation means they again become invisible. They are not visible. Remember, in telophase, they at the same time when nuclear membrane is forming, they become invisible. Their identity is lost. I made you write that one sentence, right? Transcription from chromosomes happen in the G2 phase because transcription will happen to make RNA and RNA will make protein. Protein synthesis happens in G2 phase. Okay. Replication happens in S phase. So replication is not here in question, but just I'm telling. So transcription, two options have transcription, D and B. These cannot be the right answer. Now left with A and C. This happens almost around the same time when the nuclear envelope reappear. So formation of contractile ring is just what precedes the reformation of nuclear envelope. Do you understand? It is just a very minute detail in the cell cycle. I have given you on the group that picture. You both got this picture, the picture that we discussed in the class also, the chart of all the stages of cell cycle division. You got that chart?
Yes.
I sent both the document version and the picture version. In that, if you see, so in diagram, when the nuclear envelope starts reforming, the cell also is contracting. Okay. Do you understand the logic behind the right answer? Yes? No? Iram, Aisha? Okay, very good. Okay, question number 45. Aisha, read. Who read the last question? It was Iram, right? Or was it Aisha?
Sir, I read.
Okay, Iram, read question number 45.
Meiosis to perform separation of sex chromosomes, synthesis of DNA and centromere, separation of homologous chromosomes, or separation of chromatids.
Yes, write it in the chat. Meiosis 2. See, A and B are absurd answers. I don't know why you're taking so much of time. And then you, like, it was like 50-50. You had only two options and both of you chose the wrong one. Meiosis 1 separates homologous chromosome. Last class, we just spent time on this only. First cycle, which is meiosis 1, separates chromosomes, homologous chromosomes. And then the second cycle, which is meiosis 2, is just like mitosis. It separates the sister chromatids. Yes or no? Of the same chromosome. Remember? Aisha?
Yes.
Yes, now you remember. Once I said what is the right answer. Iram, A? Separation of sex chromosomes. What? No. Sex chromosomes are just like any other normal chromosome. They're not anything special when it comes to separation of them. What happens is, let me take this question and screenshot this question. And let's go to a board to explain this. Yes. Okay. So I'm stopping this thing for a moment. And sharing a whiteboard for some time. Can you see the whiteboard, everyone?
Yes.
Let me whiteboard will not work. Let me go to the page. Yes. Can you all see this question? Yes? It's visible?
Yes, sir.
At metaphase 1, remember this was Iram's question. If it is a cell, then at metaphase 1, what are aligned at the equator? How many lines are there on the equator? 2. Right?
Yes.
2 homologous chromosomes are there on the equator, each carrying 2 sister chromatids. Yes? You understand, everyone? Both of you?
Yes, sir.
Like this. I cannot make like this. So during first meiosis 1 in the anaphase, what are separated from each other? The chromosomes, homologous chromosomes, right? But each chromosome still has 2 sister chromatids, right? So during meiosis 2, what will be separated? These 2 sister chromatids from each other. You understand? So that finally at the end, when you make 4 haploid cells, each haploid cell will have just 1 chromatid, 2 of which will be recombinants and 2 of which will be parental combination. Remember we did this in the last class?
Yes.
So in meiosis 2, separation of chromatids happen. Whereas in meiosis 1, separation of homologous chromosomes happen. Is it clear? Synthesis of DNA happens when? S phase. And separation of sex chromosome, it's an absurd because this is just like other chromosomes being segregated. Is it clear? This question is clear? Iram and Asha?
Yes, sir.
Okay, so let's start with the new chapter of today. Do you want to note this thing down? Can I delete all of this? I'll use the same page to begin the next chapter.
Question.
Can I delete this?
Yes, sir, you can delete.
Okay. Always remember that in meiosis, there is only once that the DNA gets synthesized. Very good. Okay, so what we'll do today is, let's start biomolecules. So my plan is we'll finish this whole unit 3, right? Chapters 8, 9, and 10. There are 3 chapters in this unit. So we have already done chapter 8 and 10. So let's finish with chapter 9 and finish this unit of cell, biomolecules, and cell division. How about that? Then we'll go to the beginning of classification and living systems on Earth. Okay? Good. So write down chapter 9.
Okay.
Okay, so what are biomolecules? What do you understand by the term? Let's come to the term biomolecules. So what do you mean by bio? Iram, what do you mean by bio? Biology? Bio means life, right?
Life?
Or living. Yes. And molecules are molecules, right? A group of atoms. Which make a compound. Yes. Now, these are the molecules which are special and are only present in living systems. And why do we have to study about biomolecules? Because let me ask you a question. What is the difference between if I take any non-living sample, let's say if I take a rock sample versus a human tissue sample? Okay. So in a rock sample, if I digest somehow chemically, digest everything that is present and break everything down to the level of molecules or even elements, what do you think I will find in rock? What is rock made up of? People. Carbon? Iron? Iron ores are rocks, right? And there can be calcium also in a rock. Chalk is a rock and it has calcium, right? What else can be there in a rock sample? Hydrogen? Iram, Aisha?
Yes, sir, hydrogen.
Yes. And what else can be there in a rock sample? Oxygen? Oxides? Many rocks are oxides, right? So oxygen? Yes or no? Iron oxide, ores, they are rock samples. What we will find in a human sample? Same thing, right? Carbon. Do we have iron in our muscles?
Yes.
In our
Yes.
where are we keeping iron in our body? See, if I try to confuse you, As a teacher, it's my job. It's a part of training. Don't get confused. Say, you said yes with conviction. So give the answer with the same conviction. Where is iron in our body?
Bones.
So I'm not sure, but
If our
Sir,
bones
maybe
would have,
bones.
if bones would have iron, no, Aisha? Best thing.
Sorry, no, not bones.
Yes.
so
Yeah.
but I
Yeah.
think
Yes.
it's
It's
hemo,
yes.
hemoglobin.
Yes. 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 Correct. It's hemoglobin. We have iron in our hemoglobin in the blood, right? Hemoglobin is a molecule which has an iron core. So it is said if you eat iron rich things, now you cannot go and start eating iron bars. We eat spinach and things which contain iron, pomegranate and things like that. Then you will have more blood formation in the body, right? Anemic patients, low on iron diet causes anemia. You do not make hemoglobin. So correct, you know the answer. And Asha, our bones are not made up of iron, okay? I wish they were, but they're not.
Bones are made up of calcium, right?
Yes, calcium carbonate and phosphorus. Let's come to, but same, I'll find iron, right? Correct, great. Then calcium, of course, bones. Hydrogen, yes, wherever there is carbon, to make any sugar, we have hydrogen also, right? Glucose, you know the formula for glucose? C6H12O6. This is glucose. And the structure is like this. There are 6 carbons attached to hydrogen and oxygen. Okay, so hydrogen is always there and carbon is there. And oxygen, I just told you, in glucose we have oxygen. Now let me ask you a question. How are we different from a rock then? This is a living system. This is a non-living system. It was never living. And the kind of elements we find in both are same. Then how are we different? Think logically. I know you both are logical thinkers. Do you first believe that you are different from a rock? Hiram?
Yes.
Yeah.
Yeah, very proudly. Yes, we are different from a rock. Who's a rock? Get rid of rock. Then how are you different? You just told me that you have the same things that a rock is made up of. Then how are you different? If it's not the composition, then what it can be that can make all the difference? If it's not this, see, the composition is more or less same. If you go to constituents, you have carbon, iron, calcium, hydrogen, oxygen. But if the elements are same, present in both a living system and a non-living system, then what should not be same? That should define a living system different from a non-living system. You know the answer. I know it. You just are not saying the right answer. Exactly. If it is not the composition, then it has to be the concentration of that composition, right? Yes or no? 21% oxygen present in atmosphere. You know that we believe that oxygen is good for us. Do you believe that oxygen is good for us?
Yeah.
Aisha, now I'm going to tell you a very bizarre fact. Living systems can also undergo oxygen toxicity. Too much of oxygen for the system creates oxygen toxicity. We are evolved to breathe in 21% oxygen, which is our atmospheric oxygen. When patients are at severe conditions, they are admitted to ICU, they give them oxygen masks, right? At inhalers.
Yes.
Do you think they are providing the patient with 100% oxygen, pure oxygen to breathe because you are ill, so take pure oxygen? We have a
Yeah.
myth. No. If you give 100% oxygen to someone's lung, their lungs will burn. Burn means not like start burning with flames, but will oxidize. What is burning? What is combustion? Chemistry. Just
Burning
simple
in the
chemistry.
presence of oxygen.
Perfect. Breaking down in the presence of oxygen is called combustion. Okay. We do combustion in our cells, each and every cell. We break glucose in the presence of oxygen in each and every cell of our body. That is called respiration or glycolysis. We are using the same concept of burning something outside. We are doing it in our body without flames or smoke at a very low temperature with the help of enzymes. Now, you know what are enzymes? They are catalysts. And what do catalysts do in chemistry? Catalysts make any reaction feasible and faster at that given condition, right? If a reaction is only possible at very high temperature, you give that reaction a catalyst, and that reaction can then happen on a low temperature as well, right? Yes or no? They facilitate reactions. So enzymes in our body are the same things. They are biological catalysts. So basically, if you give pure oxygen to someone to breathe, thinking that okay, ye to abhi bahut maza aayega, no. There will be so much of oxidation in the lungs that there will be tissue death, and the person will just die. So in the hospitals also, that oxygen cylinder has mixed helium with it. Helium is an inert gas. It will not react with anything in the body, and that creates, so the pressure, the partial pressure of oxygen is still around 21% in that cylinder also. But we are just putting it through a mask and giving it to the lungs through some pressure, so that the patient who cannot breathe on their own can now breathe with some help, ventilation, artificial breathing. You know, we often have a myth, you know, I've seen my parents also talking that let's go to hill stations, you know, there'll be more oxygen there to breathe. In fact, that's not true. Pure air does not mean more oxygen. You know that? A less polluted air or a clean air does not mean that it has more oxygen for you to breathe. It has the same 21% oxygen. It's just like the pollutants, other things that are harming your lungs are not present, like other particles like this 2.5 mm particles or dust particles or smoke particles, they are not present. And in fact, when you go up on hill stations, you actually are subjecting yourself to lower oxygen concentration. You know, as you go up in the atmosphere, in the altitude, you have less and less oxygen to breathe. You know that?
Yes. Yes.
Yes. So going on a hill station to breathe more oxygen is a myth. You're going on a hill station to breathe less oxygen, but just the air is less polluted because there'll be less cars and less things. And you will feel like, so much of good oxygen. No. Oxygen concentration is never more than 21% on the planet's atmosphere. Okay. So it's always the composition if it's not the, sorry, it's always the concentration if it's not the composition. And that's exactly what happens. In living systems, carbon, oxygen, hydrogen, and one more thing is nitrogen. These four things are present in abundance in living systems. These four things, there can be nitrogen in rocks also, like, but it will be very, very less in amount. Okay. Now let me pull down an image. Do you have NCERT? Please open your NCERTs if you have. Just open the chapter Biomolecules and you will see this one diagram there. Can you see this diagram? This table actually, sorry. Okay. Everyone can see this table? Both of you can see this table? Yeah? Visible to you?
Yes, sir.
Yeah. Now look at this thing. We just did the same thing. Hydrogen. So these are the elements. This is the percent weight of two options. Earth crust and human body. Look at carbon, hydrogen, oxygen, and nitrogen. Just focus on that. So this is hydrogen. How much it is in the Earth's crust? 0.14% by weight. How much it is in the human body? Also, remember that it is the lightest element, right? It just has one proton. Its atomic number is 1. Just one proton and no nothing else in the nucleus. So it's very light. Still, it is 0.5% of our human body. So can you see how much more it is? Right, 0.1 and 0.5. At least, let's say 4 times more, right? Look at carbon, 0.03 in Earth crust, because most of the carbon is in atmosphere or in the living systems. 1 molecule of glucose. Where did that glucose go? Did I remove it? Okay. C6H12O6. 1 molecule of glucose has 6 oxygen atoms. Okay. So a lot of it is in our body. So oxygen is 46.6%. Sorry, I was talking about carbon. Carbon is 0.03% in Earth crust and 18.5% in a human body. In fact, it is said that living system is carbon based. And how I can prove you? Take any living thing, and burn it. What is left after it's completely burned?
Ashes.
And what are ashes?
Carbon.
Carbon, right? So at least 18% of the body weight is carbon only. So if a human is 100 kgs and after death, if that human is burnt as a ritual, then 20%, like from that 100 kgs, around 18 kgs will be just carbon, right? You understand? So we are carbon based basically. And now look at oxygen. 46.6% it is in Earth crust, because a lot of substances are oxides, iron oxides, phosphorus oxides, all aluminum oxides, all these ores. Have you studied about ores in your class 9, 10th? I think they teach us that in geography. Remember geography?
Yeah.
Aisha is like, I don't want to remember. Please move forward. You know, geography. Okay. So geography used to ask you ores of the elements. How is copper found as copper ores, right? And most of the ores are oxides or hydrides. Now, so a lot of oxygen is trapped in the crust, in the Earth crust as oxides, 46.6%. But still it is less than human body. 65% of the human body is actually oxygen. Water, because you know that, have you heard of this thing that around 70% of our body is just water?
Yes.
Our cytoplasm has water, or all our tissues has water. We drink water, the food has water, everything, blood has water. So there's a lot of water in our body. Around 70% of the body weight is just because of water. And in that water, H2O, 18 is the weight, atomic weight, out of which 16 is just oxygen, right? And 2 is hydrogen. Yes or no? 18 or 16? 16 is water weight. Carbon, nitrogen, no, yes, 18. So oxygen, what is the atomic number of oxygen?
8.
8. So what is the atomic mass? 16, right?
16.
Yes. So that's why I was saying 16. Out of that 16, sorry, I was correct. Oxygen is 8. So oxygen's mass is 16. So total water is 18, out of which oxygen is only 16. So if 70% is water, you can just realize that where there's this 65% is coming from, right? 65% of our body is just oxygen. And then nitrogen. In the Earth crust, it is very, I'm amusing that they have written very little, because for carbon, they have written 0.03, which is, you know, very, very little. They could have written here also something, but they have written very little. Okay. So nitrogen is very little, but it is around 3.5% in our body. Okay, nitrogenous bases. All our DNA has nitrogenous bases that contains nitrogen. So if you look at these first 4 elements, you know, they are together known as CHON. This is an acronym to remember it. Remember, CHON is the basis of life. Carbon, hydrogen, oxygen, nitrogen. CHON. So write down this one line. It is not only the constitution, it is not only the composition, it is not only the composition of elements, it is not only the composition of elements that distinguish a living matter from a non-living matter. It is not only the composition of elements that distinguish a living matter from a non-living matter, but it is the relative abundance. It is the relative abundance of certain elements. It is the relative abundance of certain elements. In the bracket, write CHON, CHON, carbon, hydrogen, oxygen, nitrogen. That is higher in living matter than the non-living matter, which is higher in the living matter than in non-living matter. Is that clear? Yes? No? Clear? Hiram and Aisha?
Yes, sir.
Very well. So let's move forward. So you understood how to distinguish a living matter from a non-living matter now. And all the molecules that contains CHON in relatively more abundance are called biomolecules because they are present in living system. And biomolecules are of different types. Name some molecules for me, some biomolecules for me. Amino acids?
Proteins?
Yes, proteins. And the molecules present in proteins is amino acids, right? And lipids? Lipids are also biomolecules. Yes, Hiram.
Sugar?
Yes, third is sugar. Very good. And lipids are fats. Sugars. What are sugars basically? Carbohydrates, right? Glucose, fructose, lactose, etc. Yes. And you're forgetting the most important one. Our genetic material is made up of
Nucleic acid.
yes, nucleic acids, right? So these are most important biomolecules, and we are going to read about them in this chapter. So you understood what is biomolecules? Why are we going to read about it? Is the base clear? Yes? Shall we start?
Yes, sir.
Okay, so let's start with some basic things today for proteins, which are called amino acids. Amino acids. Now, as the name suggests, now amino acids are, just write down, they are the building blocks of every protein. They are the building blocks of every protein. And as the name suggests, amino acids, it has an amino group and it has a acid group. Acid in this case will be which acid? Carboxylic acid. Have you studied these things in your chemistry already? Tell me, because that will help me how much deep should I go, and do you already have foundations or not? Like, do you know about the amine group, aldehyde group, ketone group, carboxylic acid group? Functional
No,
groups, basically. You have not studied a functional group, Abhi,
No, sir.
in your
No.
chemistry?
no, sir.
Okay, very
It's
good.
actually deleted last year. This
It
part.
was it was deleted,
Yes.
but
Last
oh,
year.
yes, that's why I thought in class 10th, right?
Yeah, class 10th.
Yes.
This is what they do wrong. You know, when you delete that part in class 10th, and then but you do not delete biomolecules in class 11th. Either you keep deleting everything associated, right? So imagine a student who have not studied any functional group in class 10th comes to class 11th. He or she will not only struggle in chemistry, but will also struggle in biology, because they have biomolecules, which is going to use all these groups, amine group and carboxylic acid group, and then biology and chemistry teacher both will face it, will face this difficulty. Anyways, but I'll cover all the chemistry part as much as it is required for biology. We'll cover that. Okay. So remember, carboxylic acid is an organic acid. Acids are of two types. Okay, that also I have to tell. Have you studied that acids are of two types? See, Iram and Asha, I hope that both of you are okay with us spending some more time on concepts than to just finishing the syllabus, right? Because that's not my way of teaching. If you don't know something, that's not your problem. That's because syllabus, they delete something randomly from the syllabus to make their life easier. But basically they are keeping student devoid of knowledge. Then next year they will put some things which they deleted, and these things happen. So we will go from the basics. Is it okay for you?
Yes, it's okay.
Yes, sir.
Acids. You know, acids are of two types. What are acids basically? What do acids do? It's chemistry, I know, but you should know this. Acids, so you know, right? What are acids?
Yes.
Yes are not acids. What are acids? I'm like, what are acids? Iram is like, yes. You are a good question. What are acids, Iram? What do they do? They are useful, harmful. We should stay away from them.
They're harmful.
All the acids are harmful. There are two types of acids, organic and inorganic. Organic acids are not harmful. They can be, but inorganic acids are that you know are harmful ones. For example, HCl, right? Hydrochloric acid, H2SO4, sulfuric acid, HNO3, nitric acid. Do you know about these acids? Have you studied this in
Yes.
your chemistry? Yes. Okay. These are, and acids in general. What are acids? Acids are corrosive in nature. Have you studied this property of them?
Yes, we have.
Corrosive
Yes.
in nature. Yes. They do not break or degrade or burn. Again, we think it's a myth that acids burn. They don't burn, they corrode. There is a difference between burning and corroding. Corroding is like removing layer by layer the whole substance, uh, uh, breaking it down to atomic level so that everything gets corroded. Okay, so whatever substance you put acid on, if it reacts with that substance, for example, metals, they don't burn the metal or break the metal. They corrode the metal and metal is gone, broken down to pieces. So inorganic is this, they are harmful, like they corrode skin. But acids are also organic, many of which you use, many of which you eat. Citric acid. present in lemon. Okay. Citric acid is present. Have you done this one thing as a kid? I used to do this. I was a very, very notorious kid actually. My father was very, very pissed off when I used to do these things at school. If your mother gives you orange in lunch, so you will eat the orange. A normal kid will eat the orange, but a kid like me will eat the orange. But you know what we used to do with the peel of the orange? You have not done this, yeah? Hiram has done it, see? She's not just telling it because the class is being recorded. Don't worry. So if you just squeeze that, and if any of that goes in anyone's eyes, it starts burning, right? But then you can wash it. It's not harmful. It just cleans your eyes, actually. You know, now I have an excuse that I was cleaning many of my batchmates' eyes. They should, you know, clean their eyes on a regular basis. So I was just helping them. But you squeeze it and it starts burning, and then you have to go and wash it with water. Okay. So that's citric acid that's doing all of that burning sensation. Citrus fruits. The word citrus comes from citric acid. Citrus fruits has citric acid. We eat it. We also, have you heard of things like palmitic acid? If you have not, have you heard of terms like deep cleansing face wash?
Yes.
Yeah.
Not heard, probably used. They have palmitic acid. Even soaps have palmitic acids for proper good cleaning. You know, they are used. They are not very harmful. We can use it. Also,
So,
have you
but
heard of,
base,
yeah,
right?
yes, they're base, yeah. Soaps are basic, but they also have, as in palmitic acids are in face wash or these things which help you deep cleanse the skin. Okay. Or formic acid. Formic acid. How many of you have been stung by bee? Bee sting? Bee sting burns, right? It hurts. Bee sting or ant stings, they have formic acid. That's why when they sting you, there's a swelling. Yes, this one. Aisha. It's a bumblebee actually. Then you also must have heard of, I forgot, it was just lactic acid. What is lactic acid? This we eat. Lactic acid helps in curdling of milk. In milk, when you put inoculum, which is curd, that curd contains lactobacillus bacteria. They produce lactic acid, and this acid helps in acidic medium, proteins actually, what is the good word for it? They come together and bind to each other. So milk binds to each other and becomes curd. That is lactic acid. So all these are organic acids which are in living systems. These are inorganic acids which are harmful for living systems. So acids are of two types. Similarly, there is a carboxylic acid group, which is this group. If you have not studied it, it's COOH. This group is called carboxylic acid. Okay. So if you see COOH in front of any molecule, it becomes carboxylic acid. Okay. Is this clear? Just some chemistry. An amino group is this NH2. Okay, so amino acids have an amino group. Amine has nitrogen and hydrogen, and a carboxylic acid group. Is it clear? Now, amine is basic in nature. And of course, it is acid, so it will be acidic in nature. So what will be amino acid in nature? Yes.
Neutral.
Kind
Neutral?
of. In its, yes. So in its, so it shows, should I delete this? I need this space, or let it be there. Let me use this. So let's make a structure of amino acid. every amino acid, regardless of which amino acid it is. How many amino acids do we have? First, let's write down certain things which come in entrance. Facts. There are 20 amino acids in our body which make all our proteins. So it's a protein is nothing but an amino acid chain. Okay. So one amino acid bound to another amino acid like this, a long chain, and then it will fold in a particular manner. It will be called, so this is amino acid, and the bond between the two amino acids is called peptide bond. Some basics, because you have not studied this class 10. And this whole chain, because it has multiple peptide bond, it is called a poly. Poly means many. Polypeptide chain. Is it clear?
Yes, sir.
Aisha? Neeram? See, if your base foundation is done, then you will not have difficulty understanding the chapter. So a long polypeptide chain then gets folded to give rise to a protein structure. A protein is not functional until it is folded. If it is a long linear chain of polypeptide, it will not function as a protein. Is it clear, everyone? Neeram and Aisha, is it clear to you? So protein function comes from its structure, its three-dimensional structure. It does not only comes from the chain. Is it clear, Neeram and Aisha?
Yes, sir.
Okay. So there are 20 amino acids in our body, and you have to just remember physical properties of some amino acids. So write down every amino acid. Every amino acid contains an amino group, and an acidic group. An amino group and an acidic group on the same carbon. So this is the central carbon. It has on one side, it has an amino group. On the other side, it has a carboxylic acid group, but both of them are attached to the same carbon. And this carbon on which both of these are attached is known as alpha carbon. Why alpha? I'll tell you. Let's just write down alpha carbon for now. So this carbon is called the alpha carbon. Okay. Now, on one side, there will be a R group. R group. Okay. It's not, take a star here. And on the other, because how many valencies do carbon have? I think this part you have done in class 10th, right? Carbon valencies is clear to you? We are going to use a lot of chemistry in this chapter, okay? It's basically biochemistry only. So you need to brush up those concepts. You know valency?
Yes, sir.
Yeah, what is valency? Aisha, quickly. The maximum number of bonds, the maximum number of single bonds any element can form, right? So it has, carbon has 4, carbon's valency is 4. So at one side there will be hydrogen. So this is the overall structure of amino acid. Okay, very simple. A carboxylic acid group, amino group, a hydrogen, and on one side there is a R group. Now I told you there are 20 amino acids, right? This R group is the one which decides the amino acid. So this is what varies in all the 20. All these 3 things are same. Only this R group will keep differing in all the 20 amino acids. Is it clear? Yes or no?
Yes.
So write down the R group is a variable group. Questions comes on this part. R group is a variable group in the amino acid. And the nature of R group, decides the nature of amino acid. The nature of amino acid. Now, these 20 amino acids are those which make proteins in living systems. According to latest research, there have been more amino acids found, but they do not actively participate in protein synthesis, or even if they do, like it is an exception. So 20 amino acids participate in protein synthesis. So only these we have to study. We don't have to study all the 20, don't worry. Take some good breathe. It's fine. But just understand the concepts. So you understood amino acid, a central carbon known as alpha carbon, a carboxylic acid group, amino group, a hydrogen, and a R group, which is variable. And this R group will decide what kind of amino acid it will be. Okay. So this will change and the amino acid will change. Okay. And why we call this as alpha carbon? Because in the R group, there will be carbon again. Yes or no? This R group is called, what is called? Okay, I'll tell you. But it is also carbon based. So R group will have carbon, and those will be beta or gamma carbons. But the alpha, the central, is this one. Is it clear? Iram and Asha?
Yeah.
Yes. So only to the alpha carbon, all these four things are attached. Amino group, carboxylic group, hydrogen. So this was a neat question. Which of the following is true about alpha carbon in amino acid? Okay. So both amino and carboxylic group is attached to alpha carbon. Only amino group is attached always to the alpha carbon. Only carboxylic group is attached to the alpha carbon, or something, another option. But the right option is both are attached to alpha carbon. Okay. Clear?
Yeah.
Okay. Now, amino acid, as I told you, one is base and one is acid. So it has three forms. If it can oscillate between C, so let's make amino acid. So this is the alpha carbon, COOH. And if, see, the right notation is to write like this, NH3 here. Okay. And if this has a positive charge, if it is 3, then it will be positively charged. Then there is a R group and there is hydrogen. This is form A. Either amino acid can exist in this form where it has a positive charge on itself. Okay. Or, you know the meaning of this sign in chemistry, chemical reactions? Yes or no? This is like reversible state. It can go this in this direction also, and the reaction can come back also. So, or if you're for some brief moments, both negative as well as positive charge is present on amino acid. Or the third condition is where it is stable as a, please draw this, okay, yourself, negative charge. This is right, NH2. Remember to put 2 here. When it is not positively charged, it is 2. So state A, state B, and state C. amino acids oscillate, structure of amino acids can oscillate between these three states. Okay. Is it clear? Among these three states, this is a special state because it has both positive and negative charge that will cancel out for each other, still be reactive, but overall this state, the B state, is called Zwitter ionic form of amino acid. So this is a board question. Question is, what do we mean? What do you mean, or what do you understand by Zwitter ionic form of an amino acid? Zwitter ionic. Ion means simple chemistry, one-on-one. Ions. What does ion mean? Okay, tell me one more thing. Iram and Aisha, have you studied bonds in chemistry? Ionic bond, covalent bond, hydrogen
Yes, we
bond,
have.
metallic.
Yes, sir.
Okay, very good. That will help me. I would have been pissed off if that would also have been deleted. So ionic means that has a charge. A charged molecule is called ion, right? Yes or no?
Yes.
So here both the charges are there and balanced. This is called Zwitter ionic form. It is a very, very it's not very stable form. It is present for very short period of time, but it is there. Now write down. Based on which form it is more stable in. So amino acid can be stable in this form or this form, A or C. B is a short-lived in-between form. It is called intermediate form, which is called Zwitter ionic form. So write down. Okay, I was talking about this question. What do we understand by Zwitter ionic form? Write down. In a Zwitter ionic form of an amino acid, both carboxylic acid group, both the carboxylic acid group and amino group, both carboxylic acid group and amino group are in ionized state, carrying opposite charges. Okay, that's a Zwitter ionic form. Is it clear? Okay. Now write down on the basis of number of amino or carboxylic groups. So this R, do you see this R here? Now the interesting thing is this R itself can have one another amino group. Let's say this R, instead of R, let me just delete it. It is C carrying another amino group and some other carbon with oxygen or something. So you see that there is one more amino group here because this R has amino group. So now there are two amino groups and only one carboxylic group, right? You understand? Iram, Aisha, do you understand what do I mean? How can the number of amino or carboxylic group can differ? Through this R group. Because this is variable. This can be anything. It can even be benzene ring. You know about benzene ring structure?
Yes, sir. We studied.
Have you also studied that if there is a benzene ring structure, present, um, then compounds are called aromatic compounds.
Yeah.
Aromatic and non-aromatic compounds, right?
Yeah.
And why they are called aromatic compounds? Because they have an aroma. The word aromatic comes from the word aroma. Aroma means pleasant fragrance, right? Similarly, amino acids R group can also have a benzene ring or aromatic ring, because of which an amino acid can also be aromatic. And these are the things that you have to understand. So R can bring all sorts of characteristics to an amino group, to an amino acid. Okay, it can make the amino acid acidic. It can make the amino acid basic by increasing one amino group. It can make the amino acid aromatic by bringing in aromatic structure, aromatic ring. So write down, based on the number of amino and carboxylic groups, amino acids can be divided into two types. Amino acids can be divided into two types. So based on the first is acidic amino acid. Acidic amino acid. And they are named like acids. So one example is glutamic acid. Glutamic acid is an amino acid. Okay. And I'm giving you homework. Find more. Okay. After you do this homework, it's very easy. Just go on Google and find on amino acid charts where they have segregated amino acids on the basis of which are acidic, which are basic, which are aromatic, etc., which are positively charged, negatively charged, all charged. So there are charged amino acids and non-charged, like which are balanced amino acids. So you have to find more of every type. Okay, I'm just telling you types. Second is basic amino acids. Example is lysine. Yes, lysine, arginine, both. Let's say lysine. Lysine is one amino acid, which is, there are 20. It's good if you don't, I cannot force you to remember 20 amino acids name, if you don't want to, but if you can, it's good. If you cannot, just know the names, just go through it once. Okay, at least you should have heard of that name. And then there are neutral amino acids: acidic, basic, and neutral. Neutral is valine. Valine. Okay, so on the basis of number of amino acid group. Now on the basis of, so this is the first thing you have to learn about amino acid, and you have to find more of each kind. Okay, will you do this, Iram and Aisha? So this week, spend some time exploring amino acids. Okay, knowing their names, looking at the structure, especially of the aromatic amino acid. Just get familiarized with the chemistry part of it. Okay, it will also help you in chemistry. No, it's not like just for biology. And of course, in biomolecules, it will help you. So on the basis of number, there are 3 types. Second is, they can also be on the basis of ring, cyclic structure. Basis of benzene or cyclic structure in their R group. They can be aromatic or non-aromatic. So the good thing is there are only 3 acids which are aromatic. That you have to remember. Only 3. Tyrosine, tryptophan, and phenylalanine. These 3 are aromatic amino acids. Rest other 17 are non-aromatic. So you don't have to remember their name. Just remember that these 3, TTP or PTT, however you want to remember it. And let me give you one more homework. Okay, so amino acids, the way we write it, they have a 3 alphabet code. Okay, like for tyrosine, TYR is used as a 3 letter code. They also have a single letter code. If you can go and look at it, that will help you a lot. So for example, the single letter code for tyrosine, tryptophan, and phenylalanine is W, Y, and F. F for phenylalanine, Y is for tyrosine, and W is for tryptophan, I think. So W, Y, just confirm it once, and F. And the way to remember that which amino acids are aromatic, which means pleasant smelling, the acronym becomes WIFE. Okay, aromatic. Is it clear? Is it clear to you about amino acids? Yeah. So I think I have discussed with you all the major things about amino acids. What else? And then one last thing is both amino group, which is NH2, and carboxylic group, so COOH, have not the have ionizable property. Both can ionize, and that is the reason that they stay in this Zwitter ionic form. Okay. So let me stop because I'll not go into the details today. Let me stop here with the homework. First thing, find and observe. And I'll tell you a good thing. Do you have a study space, both of you? Like a study table somewhere where you can,
Yes.
or a board where you see the most, the part of your house that you see the most, an almirah or a mirror somewhere. Just take a printout of amino acid chart with structures and put it there. You don't have to remember it, but something which you can just see every time. That is a very good way. Constant visual stimulation is good way to just remember certain things, if not every detail. It's a good way. Okay, you can do that. I used to do that when I was in 10th, like in my school, basically. To find and observe amino acid chart. It's present. All sorts of charts are present on internet. I'll send a few in the group as well. Okay. Basically, what properties it should have is which are acidic amino acids, which are basic amino acids, which are neutral, and which are aromatic. Second, look at the aromatic ring structure.